Hi, my name's Courtney, and these are my group members, Sharon, Alan, and Anna. The study we're engaging in looks at how diet can modulate the gut microbiome and the role this plays in chemotherapy efficacy and the toxicity. So today we'll be discussing the ethical considerations surrounding this study and its use of an animal model.
Chemotherapy agents are often the main treatment for cancer patients and involve the use of cytotoxic drugs to kill rapidly dividing cells. Whilst effective, it results in an unwanted side effect known as mucositis due to the fast cell division in the gut lining. Chemotherapy disrupts the natural population of microflora, which influences gut inflammation as well as the activation of the immune system to suppress tumor growth. A potential prevention to mucositis development is modulating the patient's diet to alter their microbiome composition. Previous research has shown that a hydrolyzed protein diet can prevent mucositis caused by a common used chemotherapy agent, MTX. Previous research by Hannah Wardell has shown that a hydrolyzed protein diet can increase the diversity of the natural microflora and prevent mucositis. Though promising, the same mechanisms that drive chemotherapy toxicity often drives its efficacy. Thus, there is a need to determine if chemotherapy efficacy is compromised.
In order to evaluate the impact of the diet on microbiota and therefore chemo efficacy, we will utilize an animal model. As you can see, if the diet is effective, it's got the potential to improve chemotherapy efficacy as well as reducing side effects, specifically mucositis for cancer patients. Looking at the risk versus benefit scale, there is significant evidence showing that there's many more potential benefits to the human population that would result from the study versus the risks, which are mainly the unfortunate suffering of these animals.
For this study, darker gritty rats were used. So the rats arrived at the lab and were given 7 days to acclimate. After this time, 8 rats were given the test diet made of hydrolyzed protein, and the other 8 were given a regular chow or the control diet. The rats consumed this diet for 10 days before they're inoculated with the darker gritty mammary adenocarcinoma, or DAMA, subcutaneously into the flank. And this tumor is then given 4 days to grow before treatment with MTX is administered. The study then terminates 4 days post-inoculation when the peak tumor and toxicity response occur, and this is then repeated once more. So to decrease the influence of conflicting interests on the study, the scientists are actually blinded to the diet components, and they're also blinded to which animals have consumed the study diet and which have consumed the regular chow. MTX was the chemotherapy agent of choice, as it's unethical to select a drug that's not effective against a specific tumor or the specific tumor used, or is not in the patient setting just yet. So DAMA was specifically chosen in this study design as it's isogenic to the rats. It spontaneously occurred in the 1970s, and the tumor does not cause unexpected effects or discomfort, is well defined and does not metastasize, so the animal does not have to be immunosuppressed to receive inoculation.
Given that this is primarily an animal study, the Australian Code for the Care and Use of Animals for Scientific Purposes, as outlined by the National Health and Medical Research Council, is referred to in guiding the decisions regarding experimental design and execution. This involves using animals only when it is justified, supporting the well-being of animals involved, avoiding pain and distress in these animals, and applying replacement, reduction, and refinement, or the 3 Rs, at all stages of animal care and use.
This section examines the plausibility of replacing this animal model with other models where possible. It was considered whether the in vivo animal model could be replaced with an in vitro cell culture. However, the study considers multi-organ interactions. It cannot be replicated by a simple cell culture. Additionally, the use of rats as opposed to other animal models or humans was considered. Current knowledge is still premature, and hence it is not ethical to test on humans. Due to the access to DAMA isogenic tumors that originate from the darker gritty rats, it was not plausible to select a different animal model. The theory behind studying a tumor that is not isogenic to a species decreases the translatability of findings into patient settings, since tumors are isogenic to patients only.
Reduction is the ethical principle that the number of animals used in an experiment should be the lowest possible for which meaningful results can be obtained. This is done by calculating the sample size required, reducing the number of test groups necessary, and by obtaining as much information as possible from each animal used. Calculating the required sample size is important, as the number of animals used must be enough to produce statistically relevant results, wherein an observed difference in the experiment can be considered a true difference and not one that occurred by chance due to natural variation. Using too few animals is unethical, as it negates the scientific merit of undergoing the experiment because ample conclusions cannot be drawn from the data. While using animals when more than needed is unethical, as it results in the unnecessary sacrifice of animal life. With that, statistical tests are undertaken to provide a p-value to inform statistical significance. The p-value is the probability that the distributions of observed values are not different between two compared groups. It is thus generally accepted that when a p-value of below 0.05 is obtained, that the data is statistically significant. In the previous experiments surrounding this diet, using 8 rats for each group was deemed sufficient to demonstrate a statistically significant improvement in the key outcome measure. Thus, 8 rats were chosen for each group in this follow-up study as well.
So to reduce the number of animals required to produce sufficient data and to answer the research question was something that was really carefully considered. Researchers were able to achieve this by omitting study groups that had sufficient pre-existing data. So these were marked as redundant and then removed. This is groups such as control groups with no tumour. This reduced the total number down to 4 required animal groups. Additionally, another ethical consideration was obtaining as much information as possible from the same number of animals. So researchers were able to achieve this by making the study longitudinal in nature. Data in many forms was actually collected from the same animal throughout the study. This included repeated collection of biospecimens such as weight, size, tumour size, blood samples, etc. And this produced more consistent results that are robust and produced more consistent and robust findings as it decreased the discrepancy that's caused between different animals and their individual, unique features.
Refinement is classified as a method that alleviates or minimizes potential pain and distress, as well as enhancing animal well-being. There were many ways this study was refined, and we divided it into 3 subcategories to better understand them. First of all, treatment. MTX is a mild to moderate chemotherapy agent, which is self-limiting, meaning that adverse effects are reduced. In addition, care protocols for invasive procedures, such as MTX injection, were done to reduce pain, specifically using anesthesia. Moving on to supportive care, the animals have a 7-day acclimatization period to laboratory conditions, as well as physical handling by the lab managers and supervisors. Over the course of the study, handling of rats is minimized, especially post-chemo, to minimize their distress. Because chemo tends to have side effects such as decreased appetite and weight loss, the mice are provided with softened chow to ease food consumption. Also, subcutaneous water injections prevent dehydration following chemo. Heat pads are given to the rats for improved comfort and to ensure their body temperature is maintained despite the lack of mobility that they would experience as a side effect. The rats are housed in separate clay cages, which enables them to communicate and remain visible to one another, which negates loneliness, as they are social animals in nature. Due to their evolutionary fear of aerial predators, they also tend to like hiding in dark, tight spaces. Therefore, they were provided with toilet rolls, crinkle nests, and other stuff to increase their comfort. One of the most important refinement methods was establishing a clear experimental endpoint. These are classified by clinical welfare scores, giving according to different behaviors such as coat grooming behavior, movement, body weight loss, tumor size, as well as other criteria. If any animal distress is evident, the rat is euthanized. This is done to reduce prolonged suffering of the animals.
Overall, the 3 R's place, produce, refine are of great importance in ethically conducting an animal model study and ensures scientific merit whilst respecting animal well-being. The use of an in vivo animal model is designed to increase translational rigor, and with this, it is seen that the potential benefits and the value of the study towards improving cancer treatment outweighs the ethical risks that are inflicted onto animals, thus justifying use. Thank you for listening.
Thank you for listening.